I'm a teacher from University of Agadir. Today I will present a work of niche modeling, ecological niche modeling. It's a work of our colleague El-Bahi Abderafi, which could not present today, so I will do it for him. It is a special modeling of habitat suitability for Crocodylus succus in Lower Daraa Valley, Morocco. So Morocco's freshwater ecosystem in arid environments and among the most fragile and understudied habitat. Yet they once supported remarkable top predators such as Crocodylus or the West African crocodile, Crocodylus succus. This species was recorded in North Africa before in the Maghrebian region. Actually, it's extant in Morocco since 1950, and the last range in Morocco, it was in Daraa Valley. It was in some permanent water bodies called Gelta, which we call Gelta. So a growing interest in species in reintroduction and ecosystem restoration has raised an important question, which is, could suitable habitats for Crocodylus succus still exist in Morocco today? So to respond to this question, we performed the ecological niche modeling, and we focus on the Daraa Valley, which is the candidate, the last candidate habitat for the Crocodylus on Morocco. So the main objective of this study is to model the spatial distribution of suitable habitats for the species within the Lower Daraa basin in Morocco, and to determine the key environmental factor influencing habitat suitability, and finally to identify and map across within the highest potential environmental suitability for Crocodylus succus. So the Daraa Valley is it's a valley, the longest valley in Morocco, in the south of the Morocco, take place from the high mountains or high Atlas mountains to the ocean, part in the west, and crossed by the longest river in Morocco, which is the Daraa River. For climatic conditions, so the region is the temperature, or the temperature seasonality is very different from the west to the east, and the annual mean temperature can reach, it's hot and can reach 24 degrees. For precipitation, as arid region, it's receive less than 100 millimeters per year, and also the precipitation seasonality is different and very high in the coastal region. So the region or the valley is low, it's like a lowland, but in the north we have some high mountains which can reach 2,500 meters. So it is also like a bare ground with very small patches of vegetation, and which is limited to permanent water bodies or to the high elevation. We see here for the hydrological variables, so we see that we have a complex hydrological network, which is the main river. It's, as I said, it's Daraa River, but with a very small patches of water bodies or water permanent bodies. So for the environmental niche, ecological niche modelings, here the initial set of variables, the 24, and after the correlation analysis, we select the sixth, here the sixth variables, which are not correlated, which mainly related to precipitation, temperature, and distance to water bodies, and also vegetation. So we use the occurrence data that we have, which are the fourth historical recorded records, which is dispersal within the river, the main river. And for the creating models, we use Maxent, this technical issues. So for the results, the area under curve presents excellent predictive performance levels with perfect agreements also, and the main factor explaining the total variance and affecting suitable habitat suitability models is the, as you see here, the distance of to the water bodies. But also, in addition to the distance to other water bodies, we have also the distance to stream network, which is the same a little bit, and also some parameters which is related to the temperature. As we see here in the model of the habitat suitability, we see that the main suitable habitats for the presence of the crocodile, or which permit to survive of the species, it's like just to narrow and very narrow patches and fragmented, not connected, and the main are in the coastal part, and also in the coastal part, and also in the northeast part. And now the suitable area, or the suitable area, it presents just 0.35% of the total of the study area, which is very low, or very small, because of the aridity and the rarity of water bodies. Now we will see how the suitable habitats look like in reality. So here in Tantan, in the coastal area, this is the coastal area. It's near the mouth of the sea, the ocean, sorry, and this is the permanent water bodies. In Tiglit also, where this is important area because the river crossed the mountains, and we have some patches, some water bodies, some permanent water bodies, but we have also the barrier of the mountains, and also some in Tisent province or region, and also in Tata region, Aka and Ait Obli region. Donc here, how they looks like in the reality. For the proximity to the settlement, human settlement, so as we see, most of the suitable habitats are most of all are near human structure, but we have some far where the crocodile can survive. In conclusion, we create a fine-scale habitat suitability map for the crocodile sulcus within low Dar'a basin. We identify the key environmental variables which the most influencing the prediction of the species ecological niche within excellent predictive performance. Also, the habitat suitability is strongly driven by hydrographical factors. The resulting suitability map identify several areas within low Dar'a basin that may support reintroduction efforts. This finding provide a special explicit framework to guide conservation planning and reintroduction strategies for crocodile sulcus in Morocco. However, further investigation are required to assess habitat quality, ecological integrity, and long-term water dynamics and candidate state, as well as to compare them with analogous fresh water systems in Sahara Desert region. And thank you for your attention.
Okay, thank you, Aziza, for your nice presentation and also keeping time. I will invite Phoebe to present us his work from Nepal, title, come and see. Stop this one. Post-release monitoring using telemetry. Is time from Garial, head starting in Nepal. You have the floor.
Thank you very much, Natalie. I love this microphone. I feel ready to do karaoke. I won't put you all through that. Okay, so head starting and Gurial. So to start off, just a little bit of background on head starting. So what is head starting? So it's a type of reinforcement. So this is typically when you have an existing wild population and animals are released into that have spent some period of time in captivity. Specifically with head starting, this tends to mean that eggs or hatchlings are reared for some time in captivity until it is felt they're a more predator-proof size, at which point they're re-released back into the wild population. And this is based on several key assumptions. Firstly, that bigger animals are going to have higher survival due to that reduced predation, that those captive animals, once released, will be able to function like wild residents, and therefore, overall, that your head-started individuals are more likely to become mature adults in that population than those just left to hatch out in the wild. Very specifically, one of the really key things with head starting is to do some form of post-release monitoring to see whether or not it's working. I'm going to talk specifically in this presentation about individual post-release monitoring, because this enables you to understand whether a program is doing well or is perhaps not doing so well. And this is specifically recommended in the IUCN guidelines. Telemetry combined with direct observation can be a particularly useful way to do that. And what we found is that this can reveal the underlying processes leading to success or failure before you would be able to detect this at the population level. So, just a bit of background on the overall study system. So, head starting in Gurial, something that has been going on for 50 years. So in the 1970s, both Nepal and India instigated big-scale reintroduction and head starting programs for Gurial. By the early 2000s, nearly 7,000 Gurial had been released, primarily in the Ganges Basin across both countries. And typically, this didn't lead to much population recovery. To give a specific example, in Chitwan in Nepal, by 2021, nearly 1,400 Gurial had been released. And we estimated that the adult population was probably somewhere around 35. So not a great impact, and at this point it was unclear what had happened to the missing 1,200 gharial. This is particularly pertinent right now in 2026 because India is launching a new Project Gharial, and Nepal is also doing a new Gharial action plan, and it is very likely that head starting is going to continue to be an integral component of both of those programs. So the key question is there evidence that head starting is a useful tool for gharial population recovery? There are other reasons to do head starting. I'm going to focus specifically on population recovery. And if so, when and where is it useful, and how can it be done well? So looking back on 50 years of data to see different programs and whether or not they worked, we find there's 6 which we can quite clearly see didn't work. There are 3 where there was insufficient post-release monitoring to be sure whether the population recovery that was seen could be linked to head starting or whether it would have happened without the head starting. And there are 3 programs where it's still too early to tell. They were recently initiated. And one, which is the half success, half failure circle, is the one in Chitwan. So for the first 30 years, it was definitely a failure. The population dwindled to what was probably 3 adults. But since 2010, the population has shown a really steady recovery, and the majority of individuals did originate in the head starting program. This is the current distribution of gharial throughout their range. They're now limited to just the Ganges Basin, and in these isolated populations, of which the Chambal, which Jay will talk more about later, is the only self-sustaining population. And my study is up here in Nepal on the Narayani Rapti river system. So, what did we do with the specific gharial I'll talk about today? We released 20 gharial in 2019, and we used VHF telemetry to monitor them really closely over the next 2.5 years until all the tags failed. What did we find? So, just to give you a bit of context, this is the study system. We have the large Narayani River, which flows through a barrage at the India-Nepal border, at which point it changes name to become the Gandak River, but it is the same river. And its major lowland tributary, the Rapti River. To the south is the Chitwan National Park, a huge and very well-protected wilderness area. And above that is a sort of agricultural urban matrix, and in the far upstream is Nepal's fastest growing metropolitan area, the city of Bharatpur. And each of our gharial, once released, went downstream, and we essentially ended up with a set of location data for where they'd ended up, and then observations of what they seemed to be doing there. So what did we find? We found initially they all dispersed downstream. 2 eventually did go back upstream, the 2 on the bottom, but essentially we had this extended exploratory phase where they all moved downstream from the release site. This is quite important because the previous release site was just upstream of the barrage, which would have meant the majority of individuals for the first 30 years of the program all went straight into the Gandak River in India. Whereas with the new release site, probably a minority of individuals released, still maybe 20 or so per year, but not so many head down to India. And these findings support research from early on in head starting, which found that these big individuals, once released, just kind of like head off into the river system. So this is typically what you see, is you just see this increasing distance from release site, generally tapering off with time. We had one that went over 1,000 kilometers in 30 days, which was pretty impressive for a small gharial. So what we found was in this exploratory phase, we had really high mortality. So these are 6 apparent mortalities. So 4 gharial that we think died, and 2 we're pretty certain dispersed over the border, but again, were not detected by us or our collaborators in India again. Just to zoom in on what those little graphs are, I'll have a few coming up. They're essentially, I'm plotting a distance from release site, and then the points change to green when the animals seem to have established a residency. This is very typical in the literature for translocated individuals is that during this exploratory phase, you also have an acclimation period where mortality is really high before ideally it will dip back down to the baseline mortality for that demographic class. After that, we would hope to see an establishment phase, and we saw this for 4 individuals on the Rapti River. So they established a residency just after their first monsoon, or even during their first monsoon, on the river where they were released. So we kind of established this because the dispersal distance tapered off and their movement frequency really declined, which suggested they were establishing a residency. And then we found that in the Naraini, individuals established this residency much later on, typically after the monsoon rains had declined, but we had another 6 establish a residency here. Probably this was later because the Naraini is a much more dynamic glacier-fed river. And eventually reaching a settlement phase where they seemed to establish a residency where they then stayed for the rest of the period or until they died. We had a couple that shift phases in their second monsoon because we get these massive erosion events where we use whole river islands or sections. So some individuals shifted after that, but typically we found many individuals reached a settlement phase. I'm running low on time, so I'll skip this. But what we did find is other individuals who continued to move throughout the river system and during the 2.5 years of monitoring never established any form of residency while continuously losing condition. And what we think is they're essentially staying in this exploratory phase and not ending up in the settlement phase that they would have established residency. And we then looked at whether this was predictable from various measures of morphometrics at release. And we found that actually the individuals that did not establish residency were typically larger, both in terms of sort of overall size measures, but also in terms of relative size at age, suggesting they'd grown faster in captivity. And what was also interesting is that when we looked at the dead or the early death individuals, we found that there was no protection from mortality by being bigger. Probably because almost all our mortality is through bycatch in fisheries, and so it's not predation events, so being bigger is actually potentially making them more vulnerable to getting entangled in nets and killed by fishermen. So we kind of plotted this out of how they ended up at the end of the 2.5 years. We had some that continued to die even after establishing residency, again as bycatch. And we found that this anthropogenic mortality that had caused the original population decline remained the major issue going forward. And so if we want to have successful head starting in Chitwan, we ideally want the individuals to remain in the protected area where we release them, so the top 2 resident categories, but also to get rid of those dead and lost anthropogenic mortality categories that are undermining the success by continuously removing individuals. This is then just mapping out again that for those that did establish residency, we had a mixture of ones that were alive and dead at the end, and the ones that continued to move throughout, although alive, were all in poor condition, and we've not seen them since, so we're not sure of their ultimate fate. But this is, highlighting the bycatch. So just to conclude with some key takeaways, time in captivity seems to lead to some problems, probably because there is a wild phenotype and a captive phenotype. And with developmental plasticity and selection, the longer individuals are in these environments, they get sort of pushed down one route or another. So in the wild, there's a natural environment. You have to avoid predators, find resources. There's parental care. There's also the critical element of social context and learning. A lot of the bird reintroduction literature has really been looking into this in detail, but it remains somewhat neglected for crocodilians, despite clearly complex social learning behaviors. And the captive environment is very different from that, with no prey capture, very artificial social environments, and this has been somewhat overlooked in head starting to date. Thinking forwards into the future, at each stage of the reintroduction or head starting process, these different elements should be carefully considered. And graying out the mammals here because the bird and fish examples are perhaps providing some really useful models for us to look towards, particularly as fish are in similar aquatic environments to crocodilians and also tend to have things like complex parental care. And birds, of course, the closest relatives of crocodilians. So yeah, some key takeaways is post-release monitoring is essential, and telemetry in particular is critical to help prioritize threat mitigation. In situ threats have to be removed before head starting if there's any hope of success. Pre-selection of the release strategies, for example, the site and season, is very important. Smaller and younger individuals are likely better. Jay, who's about to speak next, is going to go into this in more detail for gharial, but perhaps negating the issues of longer time in captivity. And then there's a lot of pre- and post-release social context that requires further investigation, which I encourage anyone interested in this to look into. And overall, this can all be put in an adaptive management approach in order to come up with the best conservation strategy. Just want to say this is in memory of my former advisor, Bed, who sadly passed away this year, but was a key person in this. Thank you to the whole team, to the hosts here, and our partners, funders. I'm done, sorry.
Thank you, Phoebe, for your nice presentation, and also for keeping the time. I would warmly invite...
Hello all, my name is Jailabdin.
Where are you? IT, where is the IT? Can you help me?
Oh, okay.
Hello, Jailabdin. Can you hear me?
Yeah, I can hear you. Can you guys hear me?
Okay. Thank you.
Yeah. So I think I have pre-recorded the video, so we can play the video now, and I can join the question and answer session. Hello all, my name is Jailabdin. I'm from gharial Ecology Project. My friend Phoebe talked about what's Head Start and how to measure its success. I'm going to talk about how to rethink Head Start for better success. The conventional Head Start involves in collecting eggs from the wild and keeping the hatchlings for 2 to 4 years in captivity. It also involves in investing a huge amount in the whole infrastructure, and there is no post-release monitoring. From 2018 onwards, we are collaborating with Madhya Pradesh Forest Department in tracking these conventional head starts. We have tagged around 70 head starts so far. Our study shows that there is a huge mugger predation that happens on these head starts. And also, these head starts are get drowned in illegal fishing nets. Even after a year, these head starts are having a lack of ability to establish residencyship. Here's the growth comparison of a conventional head start at its 4 month with a hatchling. Actually, it has gained 65% more in length and also gained weight of 50 grams. Actually, what this means, we will see later in the presentation. Gharial Ecology Project is nearing its 2 decades of ecological studies in Chambal. Thanks to people like Pankaj, who is circled over here in red. Actually, his interest to go out and sit and observe these animals is helped us to collect data. So what data we have collected? So during the crush time, every day he go out and count how many hatchlings survive every day, and he does it until the monsoon comes. Once the monsoon comes, there will be like high flood water, and after the monsoon he go back again out and then count how many of them are survive. And here's the snapshot of it. So every year around 15,000 hatchlings comes in Chambal, hatches in Chambal, in which 45% of them will be lost before the monsoon hits. So the remaining 55% sees the monsoon. in which 50% of them lost during the monsoon, and only 5% which survives the monsoon. The ones which survives the monsoon feeds throughout the whole monsoon, and they actually grow 3 times their original size and also gains weight 8 times their original weight. Here is a reference picture of a wild survivor. Me and Guddu are carrying these animals which we caught during October hatchling tagging session. Here's the growth comparison of a wild survived hatchling with conventional head start. Actually, it is twice the size of a conventional head start. It actually shows the ability of the wild survived hatchling to harvest the food resource at its peak time of its availability, which is monsoon. The conventional head start hatchlings take at least 15 months to reach the 4-month wild survive size. Here's a new method, fast tracking head starting. We have 2 sections, pre-release and post-release. Pre-release is basically reducing the holding time of the hatchlings from years to months, and also prepare the hatchlings to hunt its own food and to avoid predators. Here's the major cost saving. Instead of big concrete pools and structures, we have used mesh enclosures with food grade overhead tanks with their top cut off as the enclosure pens. We avoid direct interventions like shifting the eggs and probing for the eggs. All the hatchlings in a nesting colony, once they hatch, they stay together as a crush. The yolk sac is used for the first 2 to 3 weeks through basking, and the hatchlings start to feed after that. It is easy to catch hatchlings from the crush directly at the time when they begin to feed. Since they are ready to feed, we provide them with live fishes of different sizes. The fishes that are commercially available in the local market create the local economy. The surprising thing is, the animals, when they catch the fish, the time taken from capture to ingestion is only few seconds. Here you can see we are working on the fish holding facility combinedly with the forest department, and also you can see the little one, my son, is also helping us. This photo clearly shows the height of the enclosure from a human eye perspective. So this gives an unique opportunity for the animals to avoid human imprinting and habituations. Here's the video that shows the hatchlings' feeding behavior. And here's the close-up of the same. You can clearly see the time from capture to ingestion is only few seconds. We collected temperature and humidity data and also the growth rate of the hatchlings for every month. We did this along with the forest officials who got trained in collecting the data. This will give us an unique opportunity to collaborate with the frontline forest staffs who can be the committed members in futures. Actually, we also installed 24/7 security cameras to record the interactions between the hatchlings and also their feeding behaviors. After feed, temperatures are the important factors in the hatchlings husbandry. The monsoonal climatic conditions out in Chambal is actually higher ambient temperatures with high humidity. We try to mimic the same in the enclosures with the heating lamp setup. Without proper suitable temperatures, the feeding of the hatchlings will be affected, which will affect in overall growth rates. Here's the size comparison among the hatchlings that are raised in 3 different conditions: the conventional head start, the fast track, and the wild survivors. Here, the fast track actually showed 77% growth rate like the wild survivor. We have collected 30 hatchlings, and all the 30 hatchlings survived through the husbandry time, which is the pre-release stage. Now we have healthy hatchlings which can go out in the wild. So the next step is post-release. The main aim of this step is to establish resident small size gharials. Our downstream study of the small size gharial shows once they establish their residentship, they show very less movement. So we want to compare the similar kind of a study with the movement ecology of the upstream smaller size classes. The post-release monitoring involves tagging the hatchlings with a lightweight VHF tag which weighs only 5 grams. We tag all the 30 hatchlings and release them by mid-November in 3 batches. We prepared interactive dashboard website to visualize the tracking data and also monitor in real time. Before going to further slides, I like to explain about the National Chambal Sanctuary. So it's a tri-state river, and it serves as the boundary for 3 states. So here you can see the yellow color denotes Rajasthan, and the pink color denotes the Uttar Pradesh, and the purple color is the Madhya Pradesh. Actually, it acts as the boundary between Rajasthan and Madhya Pradesh in the upstream stretch here, and then once it comes from midstream to downstream, then it actually serves as the boundary from Uttar Pradesh to Madhya Pradesh. But if you see in the zoomed in version over here, this is Rajasthan, and this is the Chambal River, which comes from Rajasthan, and then once it joins the Parvati here, which actually, this is the Parvati River, and once it joins Chambal, then it became Chambal. So here the Parvati is the boundary for Rajasthan and Madhya Pradesh. Here, after it joined Chambal, then the Chambal River acts as the boundary. And here's the release site, which is very near to the confluence of the Chambal and Parvati River. So now we can orient ourselves with the location. So here's the Chambal River, and here's the Parvati which is coming in, and this is the release site. So the green one is the release site, and the red one is the current animals location, and this is the confluence. So we are going to see the summary of the resident establishment of the hatchlings using the tracking data. So we have 3 groups. I'm going to give a couple of examples from each group. The first group, what you are seeing here, is the animals which are established their residency near the release site within the 2 kilometers of the release. Here's the second group, and just for the orientation, here's the Chambal River, and here's the Parvati River, Chambal and Parvati. So this group is the animals which are established their residency upstream from the release site inside the Parvati River. And here's the summary of the third group, which is the final group, and here's the Chambal River, and this is the Parvati coming and joining Chambal here. So these animals, what they did is they moved downstream, and this animal specifically moved around 8 kilometers downstream and established their residency over here. Our tracking effort shows that the released fast-track gharials settle in the same stretches where the wild survivors are seen. They also avoiding disturbances like tourism boat or humans approach or cattle's approach like the wild ones. We also seen them feeding in the wild. Now we see the survival of these hatchlings. We had released 30 hatchlings in mid-November, and we had tracked around 21 hatchlings by the first week of March. Now we have reduced to 1/3 of the population, only 10 active hatchlings. The main threat for the survival in this stretch is human interference, which is mainly fishing. When I said fishing, I didn't say illegal fishing, because here's the interesting fact. So this is Chambal, which is coming over here, and you can see that the river is being protected from both the sides. You can see the green line is on both sides of the river, but where you see the red dotted lines here is the Rajasthan side of Parvati, which is an unprotected region. I don't know what is the reason for not protecting that during the establishment of the sanctuary. But what we seen here with our tracking data is 70% of the hatchlings actually established their residency after the release within the Parvati. So the legal fishing over here is become a very big threat for the survival of the hatchlings. Here's what our recommendation to the forest department, who are the conservation managers. And again, just orienting everybody. So this is Chambal River, this is Parvati River, and this is after the confluence, it became Chambal. And similarly here, the Chambal River, the Parvati River, after the confluence, it became Chambal. So this is the stretch where in the Rajasthan side, it is not protected. So we have actually proposed a part of the unprotected stretch to give protection status, like basically a sanctuary status with the Rajasthan Forest Department. So actually in previous years, we did the same with Kunu region also. We have animals from Chambal that are moving into Kunu and nesting over there. So we have proposed to add that particular stretch into sanctuary. Similarly, we are doing the same for the Parvati region in this particular stretch. The key takeaway of the Fast Track Head Start program is to grow the hatchlings to behave like wild ones. Simply saying, good predators but bad preys. The cost-effective budget of the pre-release enables to reallocate the resource for the post-release monitoring. This new rearing method with measurable outputs created interest among all the forest department that manages National Chambal Sanctuary. In this photo, you can see the Rajasthan State Forest Minister participated in the hatchling release function. The Rajasthan Forest Department also set a new patrolling team to mitigate the fishing problem in Parvati. Madhya Pradesh and Uttar Pradesh Forest Department are in talk with our team to implement the fast track in their states. The program that we collaborated with Madhya Pradesh Forest Department, which is called River Watch, uses the local community people to mitigate the serious problem like sand mining and illegal fishing within the sanctuary limit, can also be trained to monitor the fast track head start. This sets a best example for collaborative conservation where the forest department manages, the community support the cause, and the research and science guides the program. That takes me to the end of my presentation. I finally thank all our partners for their continuous support for our project. Finally, I like to thank all my teammates who are the pillars of the project. And thank you all for patiently listening to my talk.
Thank you for this nice presentation, and thank you also to Phoebe for helping for this presentation. Our next presentation was carried out in Cambodia by Iri Gill. Is it Iri Gill? Okay. And on reproductive success and juvenile survival in an S2 conservation breeding program. Iri, you have the floor.
Perfect. Good afternoon, everyone. Bear with us, we're nearly there. So my name's Iri. I'm General Manager for Chester Zoo, the Ectotherm General Manager. And I'm going to give you a bit of an insight today of a developing partnership between Chester Zoo and Flora and Fauna International, where our knowledge and skill exchange has supported crocodile conservation in Cambodia. So a quick introduction. The first few slides will just be a very quick overview of the project and the work that we've done over there. So just to introduce the two organisations, FFI don't need no introduction, they're a huge powerhouse. But Chester Zoo, some of you might not be familiar with Chester Zoo, we're the largest zoo in the UK. And just to highlight one of the big points on there, we've invested 1.4 million in 43 conservation projects across the world. So we really are looking at how zoos can really improve impact in conservation work. So quick overview of the actual project to date. So this critically endangered crocodilian was declared extinct in 1992. Those of you who've been around a while will recognise the name Jenny Daltrey. She was kind of spearheading a lot of this work in the early times. And it's rediscovered in 2000. A kind of government and NGO conglomerate got together and developed some protection for them. 2009, ex situ component was developed and grew with 35 pure animals that were donated from farms. 2011, the first 9 animals were released, and between 2024 and 2025, the largest release to date with 50 individuals were released, including 10 to a new site, which was Varachi National Park in the north of Cambodia. Over 15 years, 246 genetically purebred crocs were released across 19 sites. And the ambition of FFI is to have an established viable population of 10,000 animals in protected areas within Cambodia. That's their target. So here's a picture of the release. This is Han Sam, who's here with us today and will be talking after me. Give us a wave, Han. He's here. You were there 2 minutes ago. And so, yeah, that's a picture of him in 2024. So the facility itself is within a government facility. It's around 40 kilometers from the capital, Phnom Penh, and it's in the facility is called Nom Tmel, and it's a 2,000 hectare site where you've got multiple NGOs working there. So Free the Bears are working there doing the bear stuff. You've got FFI doing the croc stuff. So the facilities are in the grounds of this wildlife zoo rehabilitation centre. And there's a multitude of facilities within this for FFI. So we've got multiple housings for adults, sub-adults, juveniles, incubation rearing, and very recently outdoor incubation units. Quick overview of their current kind of stock levels. I'm not going to read it out. You can see it on the stage. With as of March, there were 4 new nests in the new kind of created breeding groups. So there'll be an update for that later in the year. Quick nod to the team. Hamsan, our project manager, is here. Joe Rose, this guy is phenomenal. He's a British guy who's moved to Cambodia to look after the breeding centre, and this guy has really immersed himself within the culture, the Khmer culture, the language, and he's just really kind of integral to that project. He speaks fluent Khmer. If you close your eyes, you actually wouldn't think he's a Brit. And so our kind of partnership developed in 2021 over COVID, where the regional director at the time, Stephen Brown, contacted us and wanted support on boosting numbers at the centre. So very quickly in early discussions, it was kind of highlighted that there were very low fertility rates, very low hatch rates, very high juvenile mortality. So very, very quickly we kind of geared all the conversations around increasing fecundity, reviewing incubation, artificial incubation techniques, and reviewing the juvenile facilities. And just having a look at where we can kind of tweak and work together on coming up with ideas of where the shortfalls are. Key activity periods, so I just wanted to know more of the biology of the animal and kind of what seasons do when and whatnot. So I went out first in 2021 during the nesting season, because I was wanting to see with my own eyes, get a feel for the environment. Animals nested before I went and nested after I left, so I never actually caught any nesting, which is frustrating. And then I was back out there again in 2024, so during the hatchling season. So increasing the fecundity stock, we reviewed the kind of adult enclosures. The closures are really nice. The facility is embedded within a forest, so it's a lot of cover, a lot of complexity for the environment, quite a lot of space. The only kind of things I was tweaking on was the fact that the ground is really hard, so the nesting opportunities for the females were quite limited. So one suggestion I'd made is let's increase the depth of this substrate so the animals have further opportunities to create these nests. The meshed enclosures, because it was a forest area, had a lot of leaf litter across the top of the enclosure, so there was very little light coming through. Not a lot of UV, and most of us know that UV is really integral to egg development, bone growth, and so we cleared the leaves off the roof. The really important thing here, I'll show you the photographs later, is that the team were on board with this. They were really kind of understanding why we were making the changes, and it was quite enlightening. Adult diet review, the diet was extremely limited. It was kind of rotated between 2 prey items. Animals were overfed, so every 4 days animals were feeding. They were being offered food on rotation. So essentially, you know, the crocs aren't doing much, they're not going to be eating a lot. So it was quite limited to just chicken. So we reviewed the diet in terms of having a more appropriate diet over a monthly basis. Known calcium source, so the females were provided with catfish heads. And so talking to my nutritionists at work, catfish heads are only actually 20% calcium. So me and Joe are driving around Cambodia trying to find a better source of calcium, and obviously the pet stores are stocking cuttlefish bone, which we all know we've used historically in reptile diets. So we've introduced a higher calcium, a higher value diets there. What else do I have? And just stocking densities, so having a look at the sub-adult groups and where we can formulate kind of breeding pods amongst themselves, just to give everything a bit more space. So regarding the clearing the roofs off the mesh ceiling, you can see in this diagram here, there's very limited light coming through. There's quite a high density of animals in there, and they're all kind of butted up to the wall to try and get warmth and UV. As soon as we removed the leaf litter from the exhibit, we threw them in the other exhibits to create the substrate depth, and the animals started basking almost immediately on the land areas. And so it was really nice for the teams to see actually something they've changed has had a great impact on these animals. Review of artificial incubation techniques. So the incubators were really, really shallow substrates. They have power failure issues, and they were quite dry. And there was a lot of incursions. These guys are really proud and sensitive about their eggs, so they were checking the eggs on a regular basis. So from my perspective, we needed to create that thermal stability. So I went back to basics. I bred crocodilians before, and my techniques were refined from something I got learned from Nick Whitaker, actually, in Madras, back in the early 2000s. So we used vermiculite. There's a huge horticulture business out there, so we bought vermiculite. I did a 1-to-1 ratio, explain to the teams that they need to bury the eggs. Previously, the eggs were just sat on the surface, and they don't have to do as many incursions. So we supported by providing extra heat mats and thermometers that they can monitor the temperatures without lifting the lids off and what have you, just to create that thermal stability. And to kind of further improve the stability, I added water chambers, so water bottles and packages into the incubators themselves. So if there are power failures, you've got that retention. And that's how I transport a lot of animals abroad as well. I create water buffers, so you've got that buffer. And then while I was there, I was thinking to myself, actually, if we do manage to improve all these areas, these guys are going to have a lot of eggs. So how are we going to cope with that? The existing incubation facilities were quite modest. So I dug a chamber in the ground, put an incubation chamber with the vermiculite in, and threw some data loggers in, took data loggers with me, and we put them in the ground. I'll tell you a little bit more about that later. And then looking at the juvenile rearing, the juveniles were kept in artificial conditions because, like some other farming scenarios I've seen, the hatchlings struggle outdoors and they're susceptible to fungal infections. So these, we took a massive review to these. They're using mercury vapor lamps, which is fine, but the UVIs were extremely high. The indexes were up at 16. So in the enclosures, I was getting UV indexes of 6 and 8 for the adults. So for the juveniles, it's really intense. Those temperatures were really high as well. The water was fairly shallow. The diet was limited to fish, so there's a lot of high protein pollutants going into these shallow water bodies. Densities were really high as well. 5 minutes? 5 at least, come on. And so yes, changes were made, and we improved the kind of juvenile rearing element of it. Diets were improved also, so we reduced the high protein elements of it and added invertebrates to the diet as well. So very quickly, you can see from the last few years, the success rates from 2021 before we started, 22 was the visit, and you can see just before the incubation period, how much we've improved the hatch and the success rate. That's exclusively incubation techniques and also just improving the juvenile rearing. The other stuff I spoke about before regarding improving fecundity, I think it's a longer term kind of study really, but the immediate effect's been quite impactful, as you can see from the numbers. The outdoor incubation unit, basically the data log has revealed that the ground is great, the temperature's absolutely fine, but there were issues with flooding, so that was the big concern. So what we did, we designed a outdoor chamber that had flood mitigations built into it, and that got installed in 2024. We need to test it, so probably next year, when we've got large number of animals being produced, we'll test this outdoor incubation unit, and I can feedback on how well it went. These are the new facilities that were also built in 2024. That's a 4 by 10 metre new juvenile rearing, because the numbers are increasing now, so they need extra facilities. And that's an extra breeding pond with 5.14 new breeding stock in there. And then next steps. So there's quite a lot to kind of to follow up on now. We've kind of got where we needed to go, but further support with recommended changes, looking to validate the chambers. What else we got? Expanding of the release sites, facility development, building a robust kind of veterinary management routine screening, exploring TSD. So we've got to the point where we're producing animals. We need to know now what we're producing and what's going back out into the wild. There's other actions on there as well, but I am running out of time. And then those of you who know me, I'm a big advocate for zoos and want to really highlight the value zoos can have utilising expertise for in-country projects. These are just some of the divisions zoos have that could be drawn upon to support conservation work. I'll let you have a quick read of that, but the one that isn't that obvious are construction and maintenance teams. Facilities need to be built. We've got expertise within our zoos to kind of send staff out and support. We have a member of staff that goes out to Laos every year to help support Free the Bears in their facility design. So just food for thought, just considerations there. Media interpretation, Marissa in the steering group was talking about Shark Week, and we should do a Croc Week and stuff. And media and interpretation departments can absolutely support with us, stuff like that. Okay, bit of a rush. Some acknowledgements there from FFI and the Chester Zoo teams to make this project happen. And a big shout out to Mayuk Chatterjee, who's our Central and South Asia Field Programs Coordinator. And thanks for listening.
Thank you, Iri. Thank you for your nice presentation. The next presentation is also from Cambodia. I will warmly invite Hamsa to present us the role of captive breeding facilities and IPLSC in protecting crocodile in Cambodia. The floor is yours.
Hello everyone, my name is Samhan from FFI Cambodia. My presentation is the role of the captive breeding facility and community in reintroduction Siamese crocodile in Cambodia. Siamese crocodile. Siamese crocodile is the critical endangered species list cited Appendix 1. This species is native to South Asian or Cambodia. This species considered to extend from the wild, but in 2000, FFI and the government recovering this species in the Cardamom Mountain after the recovery, FFI and the government established the Cambodia Crocodile Project. FFI established the Crocodile Cambodia Project to protect, to prevent this species from extinction in the wild. Currently, estimate in Cambodia about 350 to 400 individuals, and mostly in the Cardamom Mountain. That is the maps about the crocodile in Cambodia. They are found most in the Cardamom Mountain, and a small population in Tonle Sap and northeast Cambodia. Yeah, our approach project approach is discovery, this discovery and validity of the population Siamese crocodile through the release and capture breeding. Approach 2, the Cambodian people will approach 2 approach two establish crocodile sanctuary to ensure the crocodile protect and manage well. Approach three, all Cambodian people are aware semi-crocodile is rare and protected by law and national priority, especially for conservation. Approach four, monitoring, research and monitoring. For capture breeding, fauna and flora, establish the Siamese crocodile capture breeding in Phnom Tamau Wildlife Refuge Sanctuary, Wildlife Refuge Sanctuary in 2001. Breed the crocodile and produce purebred Siamese crocodile for the release and supply to other organizations. DNA sampling of crocodile to identify purebred Siamese crocodile for breeding and release. Stop in 2026 total more than 400 crocodile, including juvenile. Engage with the private farm to engage with the private farm, ensure that the farm contribute the crocodile for the wild conservation. Release 2046 purebred Siamese crocodile into the wild since 2011. Improve research and technical expert on reproduction, grow rate and health management. Community engagement, community managed crocodile sanctuary. I working closely with the community in the Karma Mountain to protect the key site at the crocodile in the Cardamom. Community warden, we employ 26 community warden to monitor and to monitor and patrol Siamese crocodile and their size. Community benefit, participant bring livelihood support, eco-tourist opportunity and education program, created incentive for long-term engagement. Key outcome and lesson learned. Establish the pure stock and release 246 crocodile. Maintain the healthy population of the crocodile, more than 400 crocodile for release in the future. 26 community warden regularly patrol, monitor nest and reduce threat. Found several wild nests with the successful hatchling. Government and community still commitment and support new release site. Lesson learned, community involvement guarantee sustainability, local stability, reduce stress and build ownership. Partnership are the key. Collaboration between the government, NGO and community drive long-term success. We concentrate the work from the breeding area crocodile, release crocodile, community protection and monitoring and adaptive. Yeah, this is a graph that show about the hatchling that hatch in the facility. Yeah. This graph show about the number of crocodile released every year. Yeah, this number of wild crocodile nests that hatch successful. Example, people in 2024 we successfully hatched 60, and 2025 the successful hatch 24. Yeah. Strong government support engagement. The project, FAI project have the agreement, two agreement from the government, Ministry of Environment and Ministry of Agriculture. Both ministry support the release, support the FFI work on the crocodile conservation. Yeah. We have the global media highlight about the crocodile impact in Cambodia, such as the BBC, the New York Times, and Mong Cabbage, etc. In summary, the capture breeding supply the purebred Siamese crocodile for reintroduction. Local community provide the protection and stability for long-term survival. Political support is also crucial. This approach and effort build the species a real change at the recovery. Yeah. Thank you for the partner and donor. Yeah. Thank you.
Thank you, Sam, for your nice presentation. I will invite Robert from Robert will be presenting assessing post-release success of head-started American crocodile in Dominican Republic. Robert, you have the floor. 10 minutes.
Thank you. Hello, everyone. I'm really excited to be back here at the CSG. For those who don't know me, I'm Bobby Greco. I'm a PhD candidate at Clemson University in the US. And I'm excited to update you guys on what we've been up to in Dominican Republic. So today I'll be talking to you guys about some post-release monitoring of American crocodile, Crocodylus acutus, in Dominican Republic. So between the workshop on Monday and all of my panel mates, we've gotten an extensive background on crocodilian head starts. So I'm just going to highlight a few things. Of course, the fact that we have been doing this since the 1960s, but outside of the CSG newsletters, we don't have that much literature available about head starts, with an emphasis on post-release monitoring and how successful it is. So when I was developing my PhD, my dissertation, this had my gears turning. As I was kind of formulating my dissertation, some papers that really interest me and had me thinking about what I wanted to do were some of Ruth Elsey's work with monitoring the growth and diet of animals that were raised in captivity and released, as well as Jeff Lang and his team monitoring gharial with satellite transmitters and comparing it to wild animals from the same cohorts, and looking at how they did in comparison, as well as looking at Steve Platt and his team, seeing some success with their program with their first confirmed Siamese crocodile females nesting. It made me want to do something in the system that I was working in. So thinking about head starting, of course, captive conditions are going to be removing aspects of wild conditions. That's kind of part of the goal. We're trying to remove that predation aspect. But with that, there's some other consequences to it. How much is head starting altering the wild behavior of these head started animals? This gave me an opportunity. Luckily, my collaborators and I were initiating a Head Start program in the Dominican Republic in May of 2024. We collected 25 individuals and raised them in captivity until about between 70 and 95 centimeters total length before they were released in February of 2025. These are these guys here. Had to have a mandatory bouquet of head-started animals. So the main question for me was, how successful are Head Start programs for crocodilians after looking back at all of those, all of the literature that I could find, and how to assess it. So success, of course, we can't directly measure. So what kind of things are we looking at to determine success? Matt Shirley, of course, provided a pretty good explanation of this and discussion during the workshop. So I'm not going to kind of open that up. So we can look at breeding and nesting. Of course, within the framework of a dissertation, that's not really going to work. We know crocodilians take a long time to mature, and I don't want to do a PhD for 10 years. So I looked at growth rate, movement, and survival, and I compared Head Start animals to wild crocodiles from the same cohort. The area which I study in, if you guys haven't heard a previous talk from me, is Lago Enriquillo. It is a large hypersaline lake located in southwestern Dominican Republic, right here. Here's a close-up of it. Very unique system. I could probably do this whole presentation just on how unique this system is and the crocodiles that live in it, but we could talk about that another time. So for my project, we attached satellite transmitters, specifically Druid Argos Flex 500 units, to 3 head-started animals, as well as 3 wild animals from the same nesting cohort that we had marked previously. We surgically attached these dorsally to the base of the tail, using a leather sewing owl and braided fishing line, as well as some marine epoxy just to keep the lines kind of cemented to the units. And the plan was to run these satellite transmitters for a year and collect movement and other data. Now, unfortunately, as we kind of talked about again in the workshop, these things often have issues. So of the 6 transmitters we had, 3 of them stopped working after a month of running. But fortunately, we did have 3 units that ran for 5, 6, and 7 months. These are those units here. So what we see here is the interface for Druid, Ecotopia, and we see the first month of movement from these guys. So just some background, 61 is a head-started animal, 5F is a wild animal from the same cohort, and 65 is a head-started animal. So as we can see here, 1 we released and it kind of hung out there, established itself seemingly, and the other 2 moved around a lot. Kind of in that exploratory phase as Phoebe was talking about. Now moving forward, let's look at their last month of transmission. Something to focus on, check out 65, but before doing that, you'll notice 5F kind of establishes itself, so it stops moving around so much. 61 stayed put in the same exact area, but 65 does the most interesting thing. So it hung out in its area that it was previously, and then you see it makes this big jump. So it traveled from the northern shore all the way to the southern shore, which is pretty substantial, and also interesting because we don't see crocodiles at the southern shore. It's not really favorable habitat for the crocodiles. So something interesting to think about. Again, this is a head-started animal. Before that, transmission eventually disappeared. Now, looking into some preliminary stuff that we got to do, unfortunately, I would love to give you guys the final story, but I'm going into my final field season for the dissertation and kind of closing this part of my project. So come to my next talk. But for now, based off of preliminary morphometrics, the head-started animals versus the wild animals of the same cohort, we didn't notice any significant difference between total length and mass. But a visual observation, you can clearly see this animal here on the left is a wild animal, and that is a head-started animal. So kind of like what Jay was saying, our animals in the wild are growing faster, which is not something I would have expected. And then we also looked at the diet of these animals. We look at index of relative importance, and looking here, the diet of the wild animals is primarily fish, followed by insects, in comparison, again, to the Head Start, which is looking pretty similar. So, I mean, in these instances, this is kind of what we want to see, right? Moving into the future, I'm going to be working with this Argos data that I collected. I would like to make dynamic Brownian bridge movement models to incorporate location data and temporal data to kind of make connections between those GPS locations and create these home range maps. Again, come to my next talk. And I wanted to kind of talk quickly just about a poaching incident. I won't go too deep into this. So one of the failed transmitters was because we had a poaching event. We went out to one of our usual surveys, and we happened to find one of our transmitters on the beach far away from the shoreline at our access point. Thank you, Hallie. But on our way to the beach to survey at our normal trail to get there, we had trees cut down, as you see here, and fires lit, clearly to prevent us from getting to the shore. This launched an investigation with the Ministry of the Environment. We went out multiple days in a row to try to catch these folks who did this, and in following days, we had more obstructions on our path, so they were cutting cactus and throwing it into the path. Unfortunately, we still haven't found these people, but there's been more monitoring in this area. And I do have a video of this. I can't show it now, but if you'd like to see it, come talk to me. And just in conclusion, I am working on a Head Start review. I've talked to many of you here, and thank you if you have contributed. But if you haven't, please reach out to me. I'd really appreciate it, or come talk to me. And with that, I'd like to thank all of my collaborators and the funders, as well as Jeff Lang for talking with me and providing knowledge and guidance on the telemetry knowledge he has and how I should go about it. And with that, I will open it up to the next person. Thank you.
Thank you very much. A nice presentation. The next presentation is on the outcome, challenges, and future direction of the first Jamaica-American crocodile Head Start program. Tria, you have the floor.
Thank you. Hi, everyone. 10 minutes, okay. Hi, everyone. So my name is Treya Picking. I'm from Jamaica. My affiliations are all over the place right now, but you'll kind of understand more about who I am during my presentation on Friday. I just want to thank Phoebe for inviting me to be part of this session. Head starting can somewhat be quite a sensitive topic, which I've always shied away from talking about, but here we are. So just some quick background on our crocodiles in Jamaica. We have Crocodylus acutus. It's our only crocodilian species. It's also our only, it's our largest native reptile. And typically they're quite shy and reclusive, which makes coexistence quite easy. Well, it should be much easier. And when we look at Jamaica, it's the third largest island in the Caribbean. It's around 146 miles long, and only a small portion of the island provides habitat for our crocodiles. So we mainly find them along the south coast and in pockets along the north coast in both artificial and natural wetlands. And crocodiles were seen to be quite abundant up until the 1960s, and then they started to decline, mainly due to hunting. And then they were placed on our local Wildlife Protection Act in 1971, and then CITES in 1975. And the National Environment and Planning Agency, a government entity, are the ones in charge of the management and protection of this species. causes a lot of the threats that they face is the fact that there is an ingrained fear of reptiles and amphibians in our culture. So ingrained fear towards reptiles and amphibians in Jamaica's culture. And so we have illegal hunting, from harassment and feeding to illegal captures to pollution and so on. So there are quite a few entities and individuals that are involved in crocodile conservation work in Jamaica across the island. One such entity is the Holland Bay Crocodile Sanctuary that is located in the eastern part of the island in the parish of St. Thomas. Now, it's Jamaica's only crocodile sanctuary. It was established in 2019 by Mr. Lawrence Henriquez, Jamaica's very own crocodile whisperer. He has been involved in crocodile work, Jamaican born and raised since the 1980s. And so this area is actually within a natural crocodile habitat. And his sanctuary, kind of in the middle of nowhere, you have to kind of go down a beaten path to even access the facility. But the enclosures are built within the natural wetland. So there's freshwater pools, there's native vegetation, and it provides a very enriching environment for captive crocodiles. It's completely off the grid, it's a low impact design, and the surrounding areas are mainly utilized for agriculture, with the closest community being around 1 mile away. So there's very low population density in the immediate areas. Now, the mission of the sanctuary is really to increase the protection of the American crocodile in Jamaica through rescue and rehabilitation, outreach, research, and head starting. So, and the area itself can be split into North and South Holland Bay, which comprises of various different types of wetlands. We have a mixture of rivers and canal systems that run through. Also in South Holland, we have a main irrigation channel, which was once used by the sugarcane factory that's no longer in operation. And we have quite a lot of mangrove fringe along the coast in both areas. So when Mr. Henriquez established the sanctuary, one thing he proposed to the government was a Head Start program. And this is because he had been doing monitoring in the area, or surveys in the area, since the 1980s, and he had seen the population decline based on his field observations, and he was very concerned. And what had driven this decline was illegal hunting and also habitat degradation from, of course, like the agricultural and industrial runoff into the canals. So luckily, the talks before me have kind of told you guys all about head starting, and so it really is to increase the survival rate of young crocodiles in the wild by growing them in captivity to a certain size and then releasing them. And with our Head Start protocol, it was developed by the government of Jamaica, and then the first actual Head Start facility was established at the sanctuary when it began its operation. And our 4 main stages are collection, release, post-release, and long-term monitoring. And each stage has specific guidelines which we are meant to follow to ensure the effectiveness of the program. Now, with the Head Start program itself, Lawrence actually has around up to 8 breeding females. So there's a lot of nests that happen at the sanctuary. We're able to collect the hatchlings within captivity, but then we also do have cases of nests and also hatchlings being found in the wild, which need to be relocated, and they are brought to the sanctuary and sometimes become part of the Head Start program. Now, the 3 stages of the Health Screen, which is the big event, and it's very collaborative. It's always a really, really good activity for many, many reasons, but the main stages are capture, the health assessments, and the measuring and tagging. So the capture involves, of course, going into these enclosures. They're either concrete pens or built within the natural wetlands. We have 2 different types for the Head Start crocodiles. The downside of having Head Start crocodiles in the natural pens is that they create these burrows, and it almost makes them impossible to be caught because they dig really, really deep caves. But yeah, the team will capture them, and then we will do veterinary health assessments led by a certified vet. So they'll look at the animal, check that they're healthy enough to be released, look for any abnormalities in their body condition, or look for any diseases or infections. And then at the next stage, it will be the measuring and tagging, which is of course taking biometric data, and then also scoop clipping and pit tagging them. And then once the animal is deemed healthy enough and meets the criteria, they are then released into the wild. And then what's meant to follow next is the post-release monitoring. And so the outcomes of the program have been what we assumed, what we hope to assume has been an increase in the number of crocodiles across the Holland Bay wetlands, and then also increased training and capacity in crocodile handling and management. It's, as I said, it's a very collaborative team, so we have both local and international partners. Just to name a few, we have the National Environment and Planning Agency, the Veterinary Services Division, the Hope Zoo, Jam Croc Initiative, Gatorland Global, and we also have quite a lot of local and international volunteers that take part in these health screens. Now, we've had quite a few challenges with the program. To just be quite transparent, the COVID-19 pandemic hit shortly after it was established. So the program was established in 2019, and we didn't get to do the first release until 2021. And so with that, one of the basic requirements we had for our guidelines was for crocodiles to be maximum length of 1 meter, minimum like 51 centimeters, and be in captivity for at least a year and a half, ideally. But by the time we had this first release group, there were some individuals that had been eating very good and growing very fast, and they were around 1.5 meters. So it wasn't really ideal, or it didn't go as we had planned. But we worked with it. And then another challenge we had is because of the pandemic, we weren't able to do community engagement and outreach in the areas prior to release. And of course, if you're releasing crocodiles into the wild, interactions may increase, and you want to reduce negative interactions, especially when you have conflict situations ongoing. And we also had a vision to increase monitoring in the areas, because you want to take away the kind of illegal hunting activities. And there was that vision, but unfortunately it was more or less Lawrence himself who would go out and monitor the wetlands, and so that capacity was not achieved. We did have a few conflict situations as well. There was the community nearby Amity Hall. Shortly after Hurricane Beryl hit us in 2024, there were crocodiles seen in the canals, and one was seen near the school. And parents and residents were accusing Lawrence of releasing the animals into the area on purpose. And so we had to kind of remedy the situation by, he had to go out and monitor and try and retrieve the animals, which ended up not being Head Start animals. And then we also went out and did some outreach with the school and the community, and it was more or less fine. And then last but not least, limited resources for post-monitoring. So luckily, the island-wide survey that I'll be talking about on Friday coincided with the first year of the Head Start program when the animals were released. But after that, we didn't have the funding and capacity to actually monitor the crocodiles, which we were aiming to do for at least a year, and then you would ideally introduce telemetry studies and long-term monitoring. So this was something that we missed out on. So the big question is, to what extent should Head Starting be used as a conservation strategy for crocodile conservation in Jamaica right now? Because when we look at the island-wide survey results that we now have, the population, while it may be experiencing declines in certain areas, it seems as though the population is relatively stable. I'll use that loosely, but the concern lies more with the distribution of the population across the island more than the overall abundance. And so we have to see how we can best incorporate a priority like Head Starting if it should be a priority, because right now we want to focus on outreach and education, enforcement, habitat protection, and really focus on the population that we have remaining in Jamaica. But what I can say is that we have a really good Head Start model to work with, and we have, we can learn from the outcomes and the challenges. And if it is that with the long-term monitoring that we're now doing to determine the population trends across the island to see if declines are actually happening, then we have a program that we can readily implement and implement it in an even better way. So before I finish, I also just want to acknowledge some entities that have been very supportive of the sanctuary and in turn have allowed this Head Start program to happen. The National Environment and Planning Agency, Gatorland Global, Hope Zoo, CV Chicken, Crocfest, and also Lawrence himself, who has poured a lot of his own funds into making this amazing sanctuary happen. So thank you.
Thank you, Tria, for your presentation and also for your image. Thanks so much. Our next presenter will deliver his presentation via, are you there? Alvaro?
Yes, I am here.
Where is the IC? Can you help me?
Do you hear me? Hello?
Hello?
I hear you. Do you hear me?
Yeah, we can hear you, Alvaro.
Okay, I will be sure. I can't put my presentation. Oh, let me check. No, I can't show my presentation. Something happened. Nope. It's not possible to show my presentation. Why?
Yeah, he's supposed to.
It's not available the button to. It's saying me that it's not permitted to share.
Can we share it?
Nope. Nope, you don't permit me. Álvaro. If I pull out and try to open again the Zoom, you think it's possible? Hamza, you have the presentation in your computer?
No, we don't have your presentation. We can hear you. I'm also seeing you.
Yep.
I'm asking the IC to allow you to share your presentation.
Yep. Yes. Yes.
Okay.
Let me check what date.
Have you received the email address?
Yes, I will be check what date.
Okay, send your presentation via this address.
Okay, May 4.
Email.
Yeah, I
Email
sent
address.
to Assam and with copy to Matt Shirley. May 4, yes. Okay. Let me say by chat. How I put. It's not permitted to send by the chat.
Hi, Alvaro, can you hear me?
Yes.
Yeah, so if you go to the participant window in your Zoom,
Yeah.
yeah, at the bottom of the participant window, you can see there's a button called Claim Host. At the bottom of the participant window, you can see there's
Yes.
a button called Claim Host.
Yeah, I think I will give the floor to Pablo.
Hola, hola.
Hi, Pablo. Sorry for missing your time. Okay, Pablo will be presenting the turning time gain in population recovery. Pablo, you have 10 minutes
Okay.
to
Thank
tell
you.
us your story. Thank you.
Hello, everybody. First, I would like to thank Phoebe for inviting me to present our case in Argentina, our starting case in Argentina. Please extend my thanks to Jeff and Sasha. Sasha, great. Okay. Being the last one, I will try to move, go through more quickly because some things that I was planning to talk is already done. Okay. I'm going to start with some definitions. Phoebe is already explaining some definition for her. For me, I'm taking some other definition, try to conclude in one. The head starting is a conservation tool. It protects animals during the most vulnerable stage of their life and try to release them when the possibility of survival is higher. But we have to take into account something very important. It does not replace the habitat conservation or the adult survival. I think nobody said that before. I think that is very important. I like this slide because I try to explain that in many species the highest mortality happens very early in their life. And head starting try to move individuals through the mortality bottleneck, and basically there is a kind of attempt to help animals get through the most dangerous part of the life cycle. The main goal is to release them later when they have a better chance to survive. But Phoebe made the same question. We have to be sure if we need to implement a head starting program. The head starting program is really justified. We have to be clear that if we say yes, we have to take into account 3 points. The head starting program is only a management tool, it's not a universal solution. The second one, it makes more sense when the main losses occur in the first step of their life. And the best example of the head starting successful cases were those that combine head starting with good site management, biosecurity, genetic, and long-term monitoring. Head starting is not only the same. There are different kinds. The main one is, the first one, is the origin of the animal of the eggs. They come from the wild or the captive populations. Depending on the objectives, one of the objectives was the same that Phoebe explained, is the reinforcement populations, another the rewilding or reintroduction. And that obviously depends on what kind of context that happened. Is in the context of the conservation program, the Zoo Basic Initiative, Sustainable Use System, that depends on all of that. It's not only for crocodilians. There is a conservation tool for some other species. You can see some examples in turtles. We make some experiment, some study with our Argentinian tegus. That the head starting depends also the countries and there's obviously many conditions. We can see our friends, the Cuban friends in the right side, with the example with Crocodylus rhombifer in Cuba, and the left side, the Crocodylus intermedius in Colombia. So I moved to Argentina. The Broad Snow-tailed caiman restocking program in Argentina was implemented in Santa Fe in 1990. It was part of the broader system that included regulation, protection, local participation, and long monitoring. And later appeared some other programs that similar but not equal, that including some other species, not only as Caiman latidostris, like was the first one, Proyecto Yacaré, it was the first one in Santa Fe, and the other were add some other species, work with Caiman latidostris and Caiman Yacaré. And why the Argentina case is important? There are some key reasons. It combined head starting with governance, wetland, and conservation and local incentive. It was the first program established after severe decline from poaching and habitat pressure. Its state goal included incentive for local community. This is the most important pillar of this kind of project. Until 1997, 100% of the hatchling produced through ranching were released as population reinforcement. After in 1997, Caiman latidostris was downlisted in CITES only for ranching and for Argentina populations. After that, one part of the animals for the hatchlings hatched from the egg collecting to the wild were destined to the commercial part of the program. The program also consolidated to generate one of the more important scientific hubs for Caiman latidostris in the world. Just to show some part of the program, this is our champions that we call gauchos. There is a local people that involved in identify nests and collect the eggs. Some other part, the incubations, the hatched. The other part is the raising, the facility and the control condition, very controlled condition. Basically, because in Santa Fe, one of the southern program, during the winter, the temperature is very low. So, immediately after hatched, we got the scute in the tail, like a combination of different cuts that we identify in the whole life. After 10, 12 months, when the animals have some important size, the animals were released in the same area where we collect the eggs, because for us the genetic component is really important. We try always to involve the local people, because that is very important that they need to feel part of this program. The communication is also important because we consider that is a very sensitive component of the conservation. Media and social media help to show conservation action to the society and help to build trust. Always a releasing event receive very, very positive attention. That is highly recommended. Night count and nest count. Night count and nest count are very important also tools to follow the change in abundance and population trends. In that sense, they are very essential to move some kind of assumptions to evidence. So that is very, very important. And then we have some success of indicators. Approximately 35,000 head-starting Caiman yearling were released into the wild. Some population increased 1,500% in certain areas. And we detect with the nest increasing in amount, and the animal observation, and they also increase the observation of the animal population. And one of the stronger results is the half of the breeding females in the operation zone are individuals previously reintroduced for the program. That is one of the strongest indicator of the program. And for us, the Argentinian lesson is that the head starting works best because it's part of a larger management. The incentive for conservation are very effective, very, very effective. And that allows us to generate a lot of information to sustain the sustain basis of the science. And currently, we only have 2 programs with different purposes. One program is important in terms of the trade, and the Proyecto Yacaré now it is in suspense in terms of the trade, but it is only dedicated to the research. So we are very convinced that the impact of the sustainable use is clearly positive in the Argentina Caiman population, and we are sure that we have now to sustain that we previously achieved, that we get some stable population. The amount of nests is very, very high like I was in the 90s. And now we have another challenge to sustain, as I said before, design adaptive management and try to or look for the way to get the local commitment, because, as I said before also, for us, the incentive for conservation gave us very good result. So, that's it.
Thank you, Pablo. Thank you for your presentation. Our last presenter is Alvaro. Alvaro, are you online? We have your presentation.
Perfect. Okay. I need to see the presentation in YouTube channel in order to speak.
Alvaro? Alvaro?
Yes.
Yes. we have your presentation, but it will be difficult to share it with you. But you can.
Yeah, I know.
Yeah,
But if
yeah, you can
if you
start.
put in YouTube channel,
Okay.
when you focus the
Okay.
window,
Alvaro, we want to hear from your presentation titled "How the Head Starting has contributed to the Orinoco Crocodile Conservation Program in Venezuela." You have
perfect. Okay, nice one. Next one, please.
Go ahead.
Okay, next one. Next slide. Please. Okay. We try, thank you very much. I want to start, sorry for the problems, and my apology for not be there with you and drink some beer tonight. But the idea of this incredible session, thank you very much to Phoebe Griffin for inviting me. I want to talk about our experience in Venezuela. It's like a program of India. We started in 1978 with the first captive breeding program with Orinoco crocodile in Mazaguara Ranch. The basis of the program is the action plan for the species, when the main objective is the conservation and recovery of the natural population of the Orinoco crocodile. To achieve this objective, we raised the animal for 11 months, hatchling before release or after to release to the natural habitat. The hatchling rising born in captivity or in wildlife, we collected every year with a local, rural, and indigenous community. Next one. Hello, next one. Oh. In this figure, we show in red the areas that we make a survey for the last 10 years. We recover more or less 1,000 kilometers of different rivers where the species normally habitat. And the black point is the places that we release individual from 1990 year. The next one. Next, please. In 36 years, we realized in 70 different locations and are more than 12,000 individuals that we realize into the wildlife. But how we evaluate that the Head Starting program really worked for conservation of the species? 4 new wild populations had been generated thanks to the release. One in El Frio Ranch, another one in El Cerrado Ranch, another in Garza River, and the other in Santa Rosa. But if we evaluate individual these places, in Guaritico Creek Wildlife Refuge, the first release is in 1990 with 40 Orinoco crocodiles. Between different years, we continue realizing more individuals. Close to 3,000 Orinoco crocodiles in this area. In 1996, we found the first wild nets. And in 2006, 19 nests was observed. And my team in 1990 and 2018, only one lagoon, we reported 11 nests. That showed that the population established in that area. The next one. Next slide, please. In Capanaparo River, the first release in 1991 with 10 individuals, and between different years, more than 3,500 individuals were released. And the population increased. You can see in the graphic about the abundance, increased a lot. But what happened? In 2012, born a male in Masaguara Ranch, we realized the next year, in 2013, with a size 700 millimeters, and we recaptured this animal 6 years later, and he survived. But in the other figure, is a female that born in USA. Back, please, back. It's a female that born in USA in Dallas World Aquarium. We realized and we captured this female 13 years later. And in the area that we captured this female, normally now we found new 5 nets every year. And we suspect that it's all the females that we realize that coming from Dallas World Aquarium. The next picture, please. The slide. Next. Okay. Based on the definition of the hair starting and also called rearing and release or population reinforcement is similar description that Phoebe expressed before me. The program in Venezuela is partly successful because if you see the graphic, the general abundance index in Venezuela increased. But in another part of the Venezuela, especially in the Orinoco River eastern, the population of Orinoco crocodile disappear from the natural habitat. Next one. Next slide, please. What's the ultimate challenge or what we do to success? You can read all the activity that we're doing every year, but what is the key point? Financial support. For Venezuela, for the last, I don't know, probably 3 or 4 years, we don't receive international support to our activity. That is the critical point for the program now in my country. Thank you very much. I think it's everything that I talked today. Yeah, I'm thankful for your help to make possible this presentation.
Thank you very much for your presentation. We are now at the end of all the presentation. I will kindly invite the presenter here for the panel discussion. Thank you.
Want to ask a question?
Okay, questions, please? Yeah.
I have a general question for everybody. Based on your experiences, what are the contexts where you think head starting would be most useful?
Hello? Yes, it works. I think that when, kind of summarizing, I think some of what everyone said, is when populations have got critically small, it can be a useful, somewhat short-term measure to boost those populations back to a certain level if the habitat is still there and it's good quality and sufficient, and the causes of direct mortality that cause the initial declines have been dealt with or are in the process of, being worked on and declining, and where you have people who are willing to then live alongside this recovered population. But I think it's probably not something that should be thought of as a long-term measure. The success is when you can stop. So I don't know if anyone else wants to add.
Another question?
Okay, another question.
Again, probably for everyone. Clearly, for a population to recover, it's just not a matter of putting juveniles in. You want them to grow, to mature, and restart recruitment. I noticed, Bobby, with your hatchlings, I don't know whether they're optimum size at the time they're being released or they're slow growers. Certainly in our experience, if animals are growing slow in that first year, they'll continue to grow slow when you put them out. It doesn't matter what happens. They don't become Superman overnight. It's sort of set. So obviously raising conditions, and this may go back to Jai's situation where, getting them feeding quickly and growing well, when they go out, they're already robust and ready to go. So it was more, I suppose, everyone's mentioned survival, but not getting to maturity and being able to actually breed and
Camboya fue de
population.
terror y Pablo es un trozo de huevón para cada día está peor.
Sí, Álvaro.
Thank you.
Hey, Charlie, you asked me something? I don't hear.
If I can go again, why not? I think the fitness element is really important. I think one thing we were discussing earlier is the challenge of often the funding and the timescale people are involved is is shorter and there isn't that opportunity, but where there is the long-term opportunity to then be able to say 50% of breeding females came from the program, that's kind of the goal. And I think very much, kind of as I mentioned with the gharial at the end, this time spent in captivity and influencing both the selection pressure of captivity and the impact on developmental plasticity is really important. I know I spoke to Tessa Malign from the Mubwire project before coming about their head starting work, and they found the same thing, that individuals that had been a long time in zoo conditions just caused havoc when they were released. I think they said one turned up in someone's kitchen, which is not what you want. So I think, yeah, that's really important, and then having that long-term perspective, and I think not just maybe becoming an adult, but then actually behaving as correctly as a mature individual. So we had the first time that creche guardianship by a gharial in Nepal was a previously head-started adult female, happened a few years ago, which we were very delighted about. But then at the same time, we found some of the adults that had been released when they had been really old, so like 10 years old and very stunted, have never ever started breeding. So they kind of cross some sort of threshold, and they look good, they look great, but they're just not behaving like a normal adult. Anyone else?
There we go. Sort of more of a comment. When I look at some of the stats up there, I kind of go, you know, there's room for improvement, obviously, which, you know, in terms of incubation and all the rest of it. And something that I think I just want to make as a bit of a comment. Crocodile farmers do this every single day to maximise the number of hatchlings they get out, the survivals, growth rates, and all the rest of it. Now, notwithstanding what Jai and you guys are talking about in terms of, you know, you don't necessarily want to, you know, just the captive environment does change them in their behaviours or whatever. But I really think integrating in with farmers who have expertise in husbandry, disease management, temperature control systems, all of those kind of things, working alongside farmers could actually really benefit these programs as well.
Yeah, get involved, love it.
This side,
It's growing.
questions?
Right. Yeah, it's sort of got a general comment, but it's a question to you. That head starting really started off with sea turtles, you know, and then some of the crocodile people back in the 1960s decided, well, this sounds like a good idea. But with neither sea turtles nor crocodiles at that time did anyone have any idea of the population dynamics of wild crocodiles. So it was a purely theoretical thing. You know, the graph that Pablo put up with, here's the number of eggs, here's the number of hatchlings, here's the number, assuming some sort of idealistic survival rate thing. Now, to my knowledge, I may be wrong here, I still think there's only one measure of age-specific mortality in one species of croc. The reality is that unless you know that mortality, you're going to have problems with head starting, because what happens in a depleted population is that, well, I'll turn this around. A population at carrying capacity, at least with some of our crocs, involves crocodiles eating, bigger crocodiles eating the smaller ones. So what happens, what we see is when there's hardly any crocodiles, at least one species, the hatchlings survive pretty well, maybe 50, 60%. Then the 1-year-olds survive. Everyone survives. But once you start to get 3 and 4 and 5-year-old crocs building up, their survivorship just cascades. So in that particular context, if you're going to Head Start, you're better to go in a depleted population, like the situation in Morocco. You'd be better to go bang, put a whole lot in, because there's no one to eat them. And then just stop, let it sort itself out, rather than keep doing it. Because the thing is, the survival rates change. As soon as the population starts to change, the survival rate changes. And between species, there's huge differences in those mortality rates. Like our freshwater crocodiles have 95% of hatchlings are lost in the first year. With our saltwater crocodiles at maybe 50%. And so each different species in each different context is probably different. So I don't think there's any general rule, but it's not automatically going to work in all contexts. You really got to look at what you know about survival and what you think is going to happen. If the population's really depleted, then you've probably got a good chance to boost it really quickly, like Cambodia. We've said for, I don't know, Charlie, I don't know, but at least 20 to 30 years, don't bug around releasing 1 or 2. Like, put 3,000 in, you know, little juveniles, and boost it once, because you're only going to get one opportunity to do that. So I think it's very context specific. Head starting itself is very straightforward methodically, technically, but the effectiveness of it will be very context specific in terms of the population and the species, I suspect. But I don't know whether anyone would like to comment on that.
Graham, once I heard Ruth Elsie in a presentation many years ago that in the Louisiana program they don't or they didn't release animals because they considered that they were giving food to the adults when releasing the animal just that.
I think also there's quite a lot that can be learned from some of the other literature. Something particularly, there's been a lot of work on restocking in fish, and it's exactly that. Like at very low population sizes with no natural recruitment, you can really boost a fish population. But they've done a lot of studies with like the fish sample sizes that croc people can't dream of, you know, in a pond more than we have in a whole population. And yeah, found that in kind of naturally recruiting populations, it's essentially pointless. At best, you're replacing wild individuals. So I think, yeah, this kind of context dependency and focusing only on seriously depleted populations is kind of the only way forward.
Shall I add a point to that, Fee? What you're saying? Can anybody hear me? Yeah. Hey.
Yeah, go for it, Jen.
Can I add the point to what Phoebe said?
Please.
Yeah. So it is totally context dependent, and also depends on like the situation also. Like, so for example, 10 years back, we said the same thing about like the fast tracking to the like forest departments who we are dealing with. But they are not, like, they are reluctant for like the whole idea, like, no, we are not going to rethink or anything. But like in recent days, like when we went to this forest department of Rajasthan, and they were like, oh yeah, it's pretty new, like let's rethink about it. And we kind of demonstrated it in Rajasthan. And once we demonstrated it in Rajasthan, and we are attending an official meeting in Madhya Pradesh, which is an adjacent state who is also managing the Chambal sanctuary, and they say like, why are we not doing this? Like, so can we do this also here? So it's, I think the situations also, and like one of the corporates like Astral Foundation, who is one of our partners, is kind of came forward and like gave all those water tanks to us and say like, hey, you can, if you can use it, like we can provide you with the food grade water tanks. So it's a combination of like the situation when the people started to accept that, okay, this alternative can also work, or coming out from their boxes and think that, okay, let's step out and think whether the Head Start, the traditional way, is helping out, or whether the bigger ones are working out. So I think the basic way of moving forward of any Head Start is, I'm also not saying that the fast track is going to work in future for every situation. So assess the situation and then see whether it makes sense for that situation, and then kind of move forward, like whether we want to go fast track or traditionally we want to take the eggs and incubate it. So it totally depends upon like each context, situation, and the partners who are coming along, the stakeholders who are coming along with it.
No, it's fine. It's just reinforcing everyone's point. I take your point, Graham, about sending numbers back. So with other ectotherm species that we work with at Chester Zoo, we put serious volumes back in the hope that something will take. As an example, we've sent 111,000 snails back to Bermuda. We've reversed the red on that species. I think the contextual kind of element of it is important. My concern would be the carrier capacity of the environment and the social elements of crocodilians, and just seeing where, you know, in terms of the ratios and understanding that social dynamic in that context.
But just one more point is, I think that sometimes the goals of Head Starting programs are exclusively kind of conservation population focused. So I just want to highlight something I didn't have time to mention, but is like the educational or outreach ability of having small animals that people can see or interact with is also a key feature. So in Nepal, you know, we would never think to get rid of the Head Starting facility, like it's going to be there forever, because it has a very important thing that people can see gharial up close and interact with them. So I just wanted to flag that, yeah, even if we had, you know, if you have a totally recovered population, there is this other potential benefit of doing Head Starting on a really tiny, tiny scale, just so there are some, yeah, friendly educational crocs at hand.
Thank you very much. This is the end of this third symposium. Thank you so much for your presentation and also your response. Thank you to all the participants, because we had the most, the longest section. I will leave the floor to Sally for the next step. Thank you very much.
Thank you, guys. I'm sure we could have kept going for another half hour there, but unfortunately we do have our evening event to get to. I do have one housekeeping thing first, however. Can I please have all of the speakers who are presenting over the next couple of days come up here and just have a quick chat to me? After that, though, the buses, I believe, are outside ready to leave now and take us to our 1001 night dinner. I'm not sure what that is, but I'm pretty excited.